Hi and welcome back to Free Science Lessons. By the end of this video, you should be able to describe the features of the genetic code. You should then be able to describe the stages of translation. And if you're following the AQA spec, then you should be able to describe what's meant by the genome and the proteome. Okay, we've already seen that humans have got 23 pairs of chromosomes. Now, many other organisms have got fewer chromosomes, and some organisms have got more. All of the genes in a cell is called its genome. In eukaryotes, this includes both the genes on the chromosomes and the genes in mitochondria and in chloroplasts. As we've seen, the nucleotide sequence of a gene determines the amino acid sequence of a polypeptide or protein. Now, all of the proteins produced by the genome of an organism is called the proteome. But you need to bear in mind that only a fraction of those proteins will be produced in any particular cell type. And cells can produce a different range of proteins depending on what the cell's doing. Now, in the last video, we looked at transcription, and we saw how cells produce a complementary RNA copy of the base sequence of a gene. This RNA is called messenger RNA, or mRNA for short. The mRNA moves to the cytoplasm, and the nucleotide sequence of the mRNA is used to determine the amino acid sequence of the polypeptide. Now, the key idea you need to understand is that the mRNA nucleotides are read as a series of triplets. Scientists call these triplets the genetic code. I'm showing you the genetic code here, but you don't need to learn this. Now, this looks much more complicated than it actually is. These are the shorthand versions of each of the 20 amino acids that we find in proteins. And these are the mRNA triplets which encode those amino acids. These mRNA triplets are the genetic code. Okay, now there are several key features of the genetic code that you need to learn. Firstly, most amino acids have got more than one triplet. For example, leucine has 6 triplets, and a large number of amino acids have got 4. Because of this, scientists call the genetic code a degenerate code. Secondly, the triplet code is non-overlapping. What this means is that no base is read more than once. So this is triplet 1, this is triplet 2, and this is triplet 3. Now, if the code was overlapping, then this would be triplet 1, this would be triplet 2, and this would be triplet 3. So remember that the triplet code is not overlapping. And lastly, the genetic code is universal. In other words, the same triplets encode the same amino acids in the vast majority of organisms on planet Earth. Okay, now as we said before, in translation, the mRNA nucleotide sequence is used to determine the amino acid sequence of a polypeptide. Looking at the genetic code again, we can see that one of the triplets determines where to start translating the mRNA molecule. This is called the start triplet. The start triplet also encodes the amino acid methionine. 3 triplets determine where translation stops, and these are called stop triplets. Coming up, we look at the process of translation. Okay, now remember that in translation, the nucleotide sequence of the mRNA is used to determine the amino acid sequence of a polypeptide. I'm showing you here a very short stretch of mRNA. In reality, mRNA molecules are much longer than this. Each triplet in the mRNA is called a codon. Now, a key idea you need to understand is that in order for the mRNA to be read, another type of RNA is involved. This is called transfer RNA, or tRNA, and it looks like this. Transfer RNA has got 2 important parts. At the top of the tRNA molecule, we've got a binding site for an amino acid. And at the bottom, we've got a triplet of bases called the anticodon. I'm showing you the tRNA for the amino acid methionine. Now, the key point is that the tRNA anticodon is complementary to the mRNA codon for that amino acid. Going back to our mRNA, the first codon is AUG. This is the start codon, but it's also the codon for methionine. As you can see, the anticodon on the tRNA carrying methionine is complementary to the mRNA codon for methionine. And in fact, there's a complementary tRNA for the codons encoding every amino acid. Remember that many amino acids are encoded by several triplets, and each one of these will have a corresponding transfer RNA. Okay, so let's look at the different stages of translation. Once the mRNA moves from the nucleus to the cytoplasm, the small subunit of a ribosome binds with the mRNA at the start codon. Ribosomes contain a number of different proteins. They also contain a type of RNA called ribosomal RNA. Now, a tRNA molecule with an anticodon complementary to the start codon attaches. This is held in place by hydrogen bonds between the complementary base pairs on the mRNA and the tRNA. Now, a second tRNA molecule moves into place. The anticodon on this tRNA is complementary to the second codon on the mRNA. In this mRNA, the second codon is GCG. Looking at the genetic code, this codon encodes the amino acid alanine. And as you can see, alanine is the amino acid brought in by this tRNA. Now a peptide bond is formed between the two amino acids. This is catalyzed by the enzyme peptidyl transferase, which is actually part of the ribosomal RNA molecule. And the formation of the peptide bond requires energy provided by ATP. Now the ribosome moves to the next codon and forms a peptide bond to the next amino acid, and the first tRNA molecule is released. tRNA molecules that have been released are later attached to their amino acids by enzymes in the cytoplasm. The ribosome continues moving down the mRNA, forming the polypeptide. When the ribosome comes to a stop codon, it detaches, and the polypeptide chain is released. Now I should just point out that once the first ribosome has started moving along, another ribosome can attach to the start codon and start translating. So we might see a whole line of ribosomes making their way along the mRNA. And this means that a large number of polypeptide molecules can be produced rapidly. Once the polypeptide is correctly folded, it can then carry out its function in the cell, and we've looked at protein folding in previous videos. Okay, so hopefully now you can describe the stages of translation.